Today we are dealing with the interesting topic called as marine protozoa. I am Amal BS, student of second year microbiology at Central University of Tamil Nadu. So speaking about marine protozoa single-cell eukaryotic organism means they have a true nucleus and a specialized membrane-bound organelles. They are incredibly diverse in ranging from 1 micrometer to several millimeter. Remarkably, some of the largest protozoans are deep sea xenophores, a type of foraminifera whose shells grow up to 20 centimeter in diameter. These organisms use different structures depending upon their type. Some use cilia, which are like hair-like structures that beat rhythmically to propel them. So others use flagella, a long whip-like appendages that allows them to swim, or pseudopodia, a temporal extension of their cytoplasm that helps them to crawl along the surface to capture the food. So most marine protozoa are heterotrophic, means they fed on other organisms or organic matter. The diet commonly includes bacteria, algae, and some organic particles, making them essential players in controlling microbial population. So when we come to reproduction part, like reproduction in marine protozoa are mostly asexual. The type, like typically through binary fission, where one cell divides into two identical cells. However, some protozoa are also capable of sexual reproduction, which increases the genetic diversity within the population. So speaking about ecologically, marine protozoa are extremely important because they forms a key component of the marine food web, serving as a primary source for many larger organisms. So additionally, they contribute to the nutrient cycling by breaking down of organic matter, helping them to maintain the health and productivity of the ocean's ecosystems. So here are some of the images that are displayed on the side. These are some of the images that first one is the Bellsparium japonica. It's a ciliate. Now we'll be talking about protozoa and classification. So classification of marine protozoa, they were once called as unicellular animals because they move and eat like animals. But now they are placed in the kingdom Protista. The reason is that protozoans are single cell, and each cell does everything like eating, moving, reproducing, responding to environment. So animals on the other hand are made up of cells which differential forming tissues organ to do specific jobs. So protozoan does not have tissues or organs, so they are not true animals. So they were classified in kingdom Protozoa from later. Before that it was in kingdom Animalia. So speaking about the groups, zooflagellates, they are classified into zooflagellates, amoeboids, ciliates, and sporozoans. So zooflagellates are special features. It's like a locomotion. Based on the locomotion, they have been classified like moving using one or more flagella. Like example, Noctula, Dinobryon, and in amoeboids, they move using pseudopodia, like extension of, they are called like extension of cytoplasm. Chaos, Arilla, Arcella, examples. And ciliate, they move using cilia, that it is like whole body will be covered using cilia. Strobedium, example. And like sporozoans, they are non-motilized adult parasite, like example Plasmodium. It is very rare in the sea marine protozoa. We'll be looking onto the examples of protozoans. So one of the examples is like foraminifera, so also called as forams. It is a single cell proteins that belong to the group called Rhizaria. Their name comes from the Latin word called as foramen, meaning a hole, because their shells usually have a tiny opening. The shell is also called as test. The most noticeable features of forams is their test or shell, which protects the cells and it can have an intricate shapes. So if talking about the composition of the cells, it is made up of calcium carbonate, most common material in their shell, and also agglutinated sediment particles. Some forams stick small particles from their surroundings to make their shells. And also they are made up of proteins or chitin. They are most found in primitive forms like textularia, and also they are made up of silica. Rarely some forams shells are made up of silica, but not most of the cells. And shells not only protects the cells but also help the scientists to study the past marine environment, because forams are found in fossils in sediments. So inside I have given one image, like it's the first known drawings of the foraminifera shells that was made by the Robert Hooke in 1665 in his books called as Micrographia. Using the microscope, he showed the tiny details, shapes and holes in the shells, giving the people the first look at these microscopic organisms. As I mentioned before, like foraminifera will be having cell. Not all foraminifera will have an external shell or test. So in the species, the test if present is located inside the cell membrane. The internal test have a tiny perforations like holes that allows the cell to exchange material within the surrounding through the pseudopodia. If we look onto the diagram inside, the cell structure of the forams is divided into two main parts. So one is the granular endoplasm and transparent ectoplasm. So granular endoplasm, it is the inner denser part of the cell containing organelles and food particles. And transparent ectoplasms is the outer layer from which the granular rectulose pseudopodia extends. And these pseudopodia are thin thread-like extensions, serves as a multiple purposes. For example, like locomotion, helping the cell to move, and anchoring, attaching the cell to the surface, or feeding, capturing and engulfing the food particles, excretion, and test construction, that is cell construction. The cell is supported by the microtubule cytoskeleton, which gives the structural strength and allows the pseudopodia to extend and retract quickly, enabling the cell to interact efficiently with its environment. So now in this slide we are going to look onto the life cycle of foraminiferans. So foraminiferans have two alternating generation. A haploid form, that is megalospheric, and B form is like diploid microspheric form. So in these images you see this generation reproduce alternately by sexual as well as the asexual method, forming a continuous cycle. So life stage, explaining about life cycle, gamount, it's a megalospheric haploid A form. This stage is haploid, that is n number. This gamount produces haploid gametes by mitosis by a process called as gametogenesis. So these small motile cell forms from the gamount. So these gamete fuse to form a diploid zygote. These diploid zygote represent the beginning of the diploid B form generation. It grows into the next stage called as a gamount. So a gamount is a microspheric diploid B form, as I said before. These gamount reproduce asexually through meiosis, producing many haploid young. These haploid young individuals grow into the schizoid or gamount A form. It's also called as schizoid. Okay, schizoid is like megalospheric haploid A form. These schizoid can either continuous asexually producing another megalosphere or proceed to form gametes again to start a new sexual cycle. So now we are going to talk about the contributions of foraminifera in marine ecosystem. So its primary role in food web is foraminifera are an important food source for many marine animals, including fish, molluscs, crustaceans and shorebirds. They help in transfer energy from microscopic organism to larger animals in food chain, and also they are important in nutrient cycling. They fed on bacteria, microalgae, which helps in recycling nutrients and organic matter into the ecosystem, supporting the growth of other organisms. So sediment formation and carbon sinks. After forams die, the calcium carbonate shells sink into the ocean floor. This contribute to the marine sediments like chalk and limestone. So long-term carbon storage, which helps in regulation of the global climate, and also they serves as an indicator of environmental change. So they are very sensitive to changes in the temperature, salinity, oxygen, and nutrients. So scientists use this as a bioindicator to monitor the marine pollutions, oxygen depletions, and overall the ecosystem health. And coming to the geographical and evolutionary importance, so fossil forams help scientists to study the past climate and ocean condition. Their shell contains a stable isotopes of oxygen and carbon, which provides an important information about the Asian marine environments and evolutionary history. This slide shows the next example of protozoa, that is radiolaria. So it's also known as radiozoa. It's a single cell eukaryotic organisms that are very small, usually like 0.1 to 0.2 millimeter in size, but they produce an intricate mineral skeleton that are often beautiful and glass-like. The unique features of radiolarians is the central capsule, which divides the cells into two main parts. So if you look at this thing, it is having an endoplasms as well as the ectoplasm. So endoplasm is the inner part that contains the nucleus and all most of the organelles, and ectoplasm is the outer part which contains the vacuoles and lipid droplets that helps the cell to float. Their skeletons are almost made of silica, giving them a glass-like appearance. So radiolarians are usually found in the drifting in oceans as zooplanktons, and they have a needle-like pseudopods, so like extension of the cells that supports the microtubules. These pseudopods helps them to stay afloat and capture the food, interact with the environment while drifting in the water. So, and in the we'll go to morphology of radiolaria. So the body of radiolarian is made up of two main parts. So one is the capsule. So if you look onto these images, the central part of the cells contains nucleus and the key organelles. So polypid, that is the outer functional part that helps with the feeding and growth. And these two parts are separated by the outer wall called as peripyle, and the end of the peripyle there is a mouth opening through which you can see this thing, which opening through which the food enters the cell, and mouth may be surrounded by the tentacles or cilia, which helps to capture the food. So inside the radiolarian, the polypid grows rapidly and eventually forms a colony. These colonies consist of many specialized structures, which gives the radiolarian the distinctive shape and colors. So coming about to the reproduction part, the radiolaria reproduce mainly through binary fission, where one cell divides into two identical daughter cells. Coming to the biomineralization in radiolaria, so radiolarians produces their mineralized silica skeleton through a process called as biomineralization, which is biologically controlled by the biologically controlled deposition of the minerals. So this process is highly regulated and involves both the cell internal machinery and interaction with the environment. So first is the intracellular silica deposition. The silica forms inside the cell within a specialized structure called as silica deposition vesicle. So that is SDV. This vesicle contains a silicic acid, that is SiOH4, the soluble form of silica from seawater. Inside this SDVs, the silicic acid undergoes the polymerization to become a solid silica, that is SiO2, guided by the organism's organic matrix. So these organic matrix are made up of proteins and polysaccharides and act as a template for shaping the silica skeleton. So this template determines the intricate lattice structures and radial patterns of the radiolarian skeleton. The proteins called as silicatines may help catalyze the polymerization of the silica, similar to the process that are seen in the sponges. So sponges also have this similar type of mechanisms. and the exocytosis and skeleton assembly is the next step. Once the silica is formed inside SDVs, it is transported out to the cell surface. The vesicle fuse with the plasma membrane, depositing silica into the skeletal external skeleton, and the radiolarians continuously build on the skeleton as they grow, forming the complex radial or spiny structures typically of these organisms. So this is one of the article from which I have referred the biomineralization of the radiolaria. So this article says about the unrevealing the chemistry of radiolaria, exploring elemental insight and environmental significance. They also said about the bioluminescence and also how understanding the diversity and ecology of the radiolaria. So if you want to know about more about this thing, you can just refer to this article. It is one of the good article, and now we are going to talk about the ciliate. So coming to the morphology of the ciliate, they are diverse group of single cell protozoan known for their complex cell structure and presence of cilia. So looking onto the body shape, their shape varies depending upon the species. So some are oval, some are elongated, some are spindle shaped, while some are spherical or vase-like. So they have a structure they have a pellicle. The pellicle is nothing but the cell is covered by a pellicle, a flexible, strong outer layer that maintains the cell shape while allowing it to move. And also they have a feeding structure that ciliate have an oral groove or cytostome that leads to the cell mouth, where the food is ingested and enclosed in the food vacuoles for the digestion. So they have one more important structures called as the contractile vacuoles. So they are the specialized cell structure in ciliate that pumps out excess water and ions. So this helps the cell maintains a proper balance and survive in the fresh or hypotonic environment, whereas the water naturally tends to enter the cell. Ciliate, now this slide discusses about the ciliate as bioindicators. So ciliates are widely used in monitoring the environmental health because their abundance, diversity and behavior respond quickly to the changes in condition like pollution, oxygen levels, nutrients and water quality. Since they are important in the microbial food web, any disturbance in the ecosystem such as organic pollution, heavy metals or eutrophication is quickly reflected in the climatic community. So using as a water quality indicators in sewage treatment plants, ciliate like Vorticella and Aspidisca indicate the good treatment efficiency. So conversely, dominance of crawling or carnivorous ciliates may suggest the poor water quality. So then also like pollution sensitivity. Some ciliates such as euphalotes and parameciums are very sensitive to toxins or low oxygen. So their absence can signal the pollution stress. So marine ecosystem in ocean changes in ciliate diversity or presence of the mixotrophic species like Strobidium or Tinisnopsis can indicate the nutrient enrichment, climatic change effects or shift in the phytoplankton population. So in this slide we are going to discuss about the zooflagellate. So they are the single cell protozoans that live in the marine and as well as the freshwater habitats, moist soil. So some live in the symbiotents or like parasite or inside other organisms. They are usually like 2 to 50 micrometer in size, though a few species can grow up to 100 micrometer. So coming to the morphological part, zooflagellates are typically elongated, spindle shaped or in oval shape. Their cell surface can be smooth or covered with pellicle for protection and support. So they have one or more flagella, so for the movement, so like anterior and posterior. So anterior flagellum often pulls the cells forward, and the posterior flagella helps in the steering or creating the water currents to bring the food closer. So some species have a feeding group or cytostome, which helps them to ingest bacteria and small protists. So in the side I have given images like morphology of the model of diplonimid. So it is just an example of the zooflagellates. So image shows the light and scanning electron microscopy. A shows the light and B shows the scanning electron microscopy. So looking about the cell, they have a sac-like shape when it is cultured, and it process a heterodynamic flagella that is moves differently and used for locomotion and it for feeding. So in down it shows the transmission electron microscopy. So that is longitudinal section. The cell contains a prominent nucleus as its center, and it has a single reticulated mitochondrium located in the periphery of the cell. This mitochondrium features the large distal cyste fold inside the mitochondrion, and also it has a Golgi apparatus also clearly visible, indicating active processing, transport and cellular materials. Now we are going to talk about the nutritional ecological role of the zooflagellates. So zooflagellates are primarily heterotrophic protozoa that feed on bacteria, as said. They capture the prey using the feeding group or cytostomes and engulf via phagocytosis. So enclosing the food in food vacuoles where digestion occurs. So some species are mixotrophic, that is combining the heterotrophy with photosynthesis by hosting symbiotic algae, allowing them to survive in the nutrient poor condition. Ecologically, like marine zooflagellates are the key components of microbial loop. So by feeding on bacteria and as well as the small phytoplanktons, so they reduce the or recycle the nutrients and transfer energy from microbial producers to higher trophic levels such as ciliates, copepodids and small fish larvae. This gazing helps control bacterial population, maintain water quality, support carbon cycling in marine ecosystem. So free living species like Bodo, Diplonema are one of the most important gazers in the coastal as well as the marine environment. So some symbiotic species also contribute to the nutrient acquisition and in host organisms. So these are some of the references that I have provided here. Thank you.